Good morning to one and all. A 3-day online national webinar, Analytical Methods for Stability Testing of Pharmaceuticals and Herbal Formulations. Welcome note like it given by the coordinator, that is Dr. A. Srinivasarao. A very good morning to one and all. On behalf of Sri Vishnu College of Pharmacy, I, Dr. Srinivasarao, coordinator, and Dr. N. Sylvia, organizing secretary for this webinar, would like to welcome our guest speaker for today's session, Dr. Suresh Bobosa, K, Principal Scientist, Division of Natural Products Chemistry, CSIR, Indian Institute of Chemical Technology, Hyderabad. And also welcome to Dr. K. Nataraj, Principal, and Dr. Kumar V.S. Nemani, Director, Sri Vishnu College of Pharmacy, Bhimavaram, to the day 2 of 3-day online national webinar on analytical methods for stability testing of pharmaceuticals and herbal formulations, organized by the Department of Pharmaceutical Analysis and Department of Pharmacognosy in association with IPA Education Division, Andhra Pradesh. So, we will like to show a small video clipping of the Sri Vishnu College of Pharmacy. Thank you, sir. Firstly, I would like to introduce our principal and director to the gathering. Our principal, Dr. K. Nataraj, completed his B.Pharm from Gulbarga University, Karnataka, M.Pharm from MGR University, Tamil Nadu, and PhD from JNTU, Anantapuram. He worked in various teaching designations at Chaitanya College of Pharmaceutical Education and Research, Warangal, and presently serving as principal of our college since 2020. He has about 20 years of the teaching experience. Dr. Nataraj sir published about 70 papers in reputed national and international journals. He has guided 5 PhDs and guiding 3 PhDs and several PG students. He is specialized in the research area like analytical and bioanalytical method development and stability indicating studies of APIs and their pharmaceutical formulations. He has wide research experience on several DST and ASCT research grants to his credit. I would like to introduce our director, Dr. Kumar V.S. Nemani sir, Sri Vishnu College of Pharmacy. Since 2018, holds B.Pharm and M.Pharm degree from Andhra University, Visakhapatnam. He also holds the PhD degree from Naipur, Mohali, and postdoctoral fellowship from McGill University, Montreal, Canada. Dr. Kumar was specialized in the research areas like oncology, autoimmune disorders, respiratory disorders, and pain. He has published, presented more than 70 papers in reputed Indian and international journals and conferences. Dr. Kumar V.S. Nemmani has over 14 years of industry research expertise on drug discovery, on development, and managerial experience. Now, I will like you to introduce today's speaker, Dr. Suresh Bobosa, Principal Scientist, CSR, IICT Hyderabad. Over to the Tanuja M.Pharm student. Thank you.
Good morning to one and all. I, Ms. M. Tanuja M.Pharm, with pleasure to welcome today's guest speaker, Dr. K Suresh Prabhu Garu, to talk about an application of hyphenated techniques in natural products research strategies and methods in the search for bioactives. Dr. Suresh Prabhu, sir, working as a principal scientist at IICT Hyderabad in natural products chemistry since 2007. Sir did his master's in 1999 and his PhD in natural product chemistry in 2005 from IICT, and a degree was awarded by Kakatiya University, Warangal. Sir also pursued his postdoctoral research at National Center for Natural Product Research at University of Mississippi, USA for about 3 years. During his 15 years of research, he was isolated several new bioactive molecules from natural sources and has 10 patents in his credit, which explains his excellence in research. Sir has published 170 papers in reputed journals. His research of interest are new drug discovery from natural products, herbal drugs and their standardization, botanical pesticides, chemical modification of lead compounds, and SAR studies. He was guided 20 PhD students and number of doctoral and postdoctoral students. Sir is a recipient of several awards and honors to his credit. Most important are OPPI Young Scientist Award 2010 by Organization of Pharmaceutical Products of India, OPPI, Mumbai. Young Scientist Award 2010 by Andhra Pradesh Academy of Sciences. He is an editorial board member of several reputed journals like Pharmacognosy magazine and so on. Thank you for giving me this opportunity to introduce about sir.
Now I request our speaker, Dr. Suresh Babu sir, to begin his talk. Over to you, sir.
Yeah, first of all, I would like to thanks the management committee and organizing committee for giving the opportunity to share our some of the research work in this platform, and the today I would like to wish International Women's Day to all lady faculty members and lady students. So I will share the screen, sir. 1 minute.
Okay, sir.
So is it visible, sir? Hello, Srinivasrao garu.
Sir, just open, sir, but slide share is not there.
Okay. Now
Your screen was visible.
slides are moving, sir.
No, sir, not yet.
Okay.
Mute all. Okay.
1 minute, sir. Sorry.
Okay, sir.
Now is it okay, sir?
Only your mail folder was open, sir. PowerPoint not yet open, sir.
PowerPoint I opened already.
Maybe it takes a few minutes times.
Okay.
వీళ్ళు వెయిటింగ్ లో ఉన్న వాళ్ళని ఎలా చూడాలి? ఇక్కడ చాట్ లో
సార్ ఏమన్నా ఓపెన్ అయిందా మీకు? శ్రీనివాసరావు గారు.
నో సార్.
నాకు కనిపిస్తుంది కదా.
ఆర్ యు షేర్డ్ సార్? షేర్ చేశారా?
చేశాను నేను.
వేరే విండో మీది ఇన్బాక్స్ విండో ఓపెన్ అయింది సార్. పవర్ పాయింట్ కనపడటం లేదు.
ఓకే ఓకే.
అందుకే ప్రాబ్లం లేదు సార్. సార్ జస్ట్ ఓపెన్ సార్.
ఓకే సార్. ఫుల్ స్లైడ్ మోడ్ వచ్చిందా సార్ ఇప్పుడు?
నో సార్. జస్ట్ పీపీటీ మోడ్ ఇస్ దేర్ సార్. ప్లీజ్ గో ఫర్ ద స్లైడ్ షో మోడ్.
స్లైడ్ షో చేశాను నేను.
ఓకే సార్. Anyhow, it is visible, sir. Yes, sir.
ఇప్పుడు స్లైడ్లు మూవ్ అవుతున్నాయా సార్?
Just first slide was open, sir. Okay, please start, sir. Yes, sir. Moving, sir.
Okay.
Thank you, sir.
Good morning to all. Sorry for the inconvenience. So we generally use MS Teams, so we are more familiar with that software. Zoom is very rarely used, so very sorry for that. Today I would like to give emphasis on the importance of first of all importance of the natural products, how they have been serving the mankind from the last several centuries, and in the natural product drug discovery, particularly the applications of the hyphenated techniques, UPLC, MSMS, and HPTLC. I would like to show some of our recent work regarding this UPLC, MSMS, and HPTLC based isolation and identification of some bioactive compounds from the natural products. And I would also give just a few glimpses about the standardization of the herbal formulations, and in that aspect I will also share some of our recent work regarding the standardization point of. So as we know, the medicinal plants, nature is a versatile source of chemically diverse and unique bioactive molecules, which are always beneficial to the humankind. And these medicinal plants are serving mankind from the last several centuries in the form of a traditional medicine. And how the traditional medicine has been formed. The traditional medicine has been formed in several centuries ago, where in that era the people and the different societies from the different parts of the world have identified some of the plants, medicinal plants, and some of the plants are toxic plants based on their indigenous knowledge. So that knowledge has been rooted to their culture and formed as a the traditional medicine. As such, we can call the Chinese traditional medicine, Indian traditional medicine, and South African tradition. All these the traditional medicine has been formed like that. And in India, particularly, the traditional medicine can be classified into two categories. One is a codified system and non-codified system, that is folklore system, where the majority of the tribal people will utilize this folklore medicine system. There is no scientific validity and regulatory affairs and all those. And of course, the both the systems contributing equally to the primary health care in India. So this traditional medicine, is serving the mankind from the last 4,000 or more years ago. But in this present scenario, where there is a revolutionary techniques has been evolved in biology and analytical techniques, we can do much better. The major hurdle in the natural product drugs is nothing but a quality control aspect. So in this present scenario, in this revolutionary techniques of analytical and biology, we can do much better. So in developed countries, particularly the Europe and America and all those, then the plant-based medicine complement Western medicine, where there is the larger population have their access to the retail shops, where this access to where some of the medicines, these medicines are called complementary and alternative medicines, and these are all often self-administered as a standardized medicines in the retail outlets. Here I gave some of the pictures, retail outlet shops in different countries. And however, in the developing countries and underdeveloped countries, this has been accessed by the traditional healers, or these people are called herbalists. We can come across this type of people, particularly in the rural areas or interior areas of our country, different corners of the country, and these people are scattered in the entire corner of the country, particularly in Andhra Pradesh, the Nalamala forest region. Those tribes are called Shinchu tribes. So majority of the times, most of the times, these people are depend upon the plant-based medicine, and they are also serving the surrounded people for their health care based on this plant-based extracts. And these people are called traditional healers or herbalists. So recently, Government of India also conducted a survey regarding the existence of these people. Almost 4 to 5 lakh people are existing in the entire country. Even though they are not educated, they got the experience or knowledge from their forefathers or from their fathers. So this knowledge has been continuing from the generations together, and they are serving in the tribal areas, particularly in the malaria and all these diseases, chronic diseases to non-chronic diseases, by advocating this plant-based extracts, folklore system. So as majority of the tribes has been utilized this folklore medicine, when coming to India, India has the largest population when compared to the other parts of the world. And of course, the tribal people are not homogeneous tribes, all heterogeneous tribes. That means every area there are different tribes are existing, particularly in the Nalamala forest, those people are called Shinchus. Similarly, in the Telangana and Chhattisgarh border, those people are called Gutti Koyas. Similarly, other tribes like who are existing in the Himalayan region, Northeast region, South region, Western India, and Andaman and Nicobar Islands. So there are majority of the tribes are scattered in these areas, and they are completely depend upon the plant-based folklore medicine. So based on this preliminary information, recently World Health Organization also conducted a survey regarding the utilization of the plant-based medicine, and it has been revealed that more than 80% of the population around the globe, they have been depend upon the plant-based medicine. And further, they have also identified another 21,000 medicinal plants which has been reported for medicinal uses around the world. And coming to Indian conditions, India is known as the botanical garden of the globe, and we have India has 16 agroclimatic zones and around 45,000 different medicinal plant species which are fairly utilizing in the Ayurvedic system of medicine or other system of medicine in the traditional medicine, and also some of the species were also fairly exporting to other countries. So these are the main natural sources: plant kingdom, microbial world, animal sources, marine world, and venoms and toxins. So every source has contributed a lot in the form of medicines, and we can among these, plant kingdom has served a lot in the form of traditional medicine like Ayurveda and Siddha, Unani. And microbial world, we know best example is penicillin, which has been discovered from the microbial sources, and animal sources. Also, some of the drugs has been evolved from the animal sources. Best example is epibetidine, discovered from the frog and other animals also. So marine world, you can see majority of the antibiotics and anti-cancer drugs which are in the present market has been originated from the marine world. And of course, venoms and toxins also, some of the drugs has been evolved and discovered. And the other source, lichens, this is also a not explored source, even though the lichens also has their importance in the healthcare, and also recorded and documented its use in the ancient system of medicine. And lichens are nothing but the symbiotic organism of fungi and cyanobacteria. And lichens also possesses all the activities as plants having antioxidant, cytotoxic, antiviral. And of course, lichens are used as a nutraceuticals in the Europe particularly, because the lichens contains majority of the times the polyphenolic compounds which are very well known as the antioxidant and other activities. And we can see lichens particularly when you buy some biryani material in the supermarkets that contains biryani leaf and lavanga and ilachi as well as the lichens. So particularly if you go to Rayalaseema region, you will get lichens, edible lichens in the retail outlets also as a biryani leaf material. So when coming to biodiversity and our strength, among the 34 mega diversity hotspots, our country hosts three hotspots like Indo-Tibetan region, Himalayan region, Western Ghats. Among these three sources, there is a lot of plenty of medicinal plants available. Even of course, not even identified taxonomically, but these plants are serving to the tribal people in the form of their for their health care as well as the livelihood. So this is a very good opportunity to focus on these areas, particularly in the drug discovery aspects. Why the plant-based drugs? Because plant-based drugs have the evidence of long history and also evidence. We can trace it the utilization of the plants almost to 2600 BC, where which has been reported by the utilization by the Mesopotamian civilization, and followed by the Egyptian civilization and recorded as a 1500 BC. In that time, almost 700 drugs were documented in the Egyptian pharmacopoeia Eber's papyrus, and followed by the Chinese medicine system, traditional system, Chinese Materia Medica, which can be traced way back to 1100 BC. And these are traditionally established system, Ayurveda. And Ayurveda is a long established system of medicine. Now it is getting more attention in the worldwide as an alternative system of medicine. Of course, Ayurveda speaks about the extracts and decoctions and kashayams and all those things, but it never speaks about the single compound based. Of course, this Ayurveda system became a basis for the discovery of many drugs which are in the present market. I am giving some of the lead compounds where these all the leads have been discovered from based on the Ayurvedic knowledge or traditional knowledge. Rawalpia alkaloids, which are used for reduce the hypertension, discovered from Rawalpia serpentina, and soralins, very well known in vitiligo and derived from Soralia corifolia. Similarly, Olarihenna alkaloids used for amoebiasis, derived from Olarihenna antidysenterica, and Mucina purens is known for Parkinson disease, and piperidins, very well known as a bioavailability enhancers, derived from different piper species, and picrosides, they are used for hepatic protection, derived from Picrosia curora, and very famous formulation used throughout India and other countries also. Phyllanthus are very well known as a antiviral agents derived from different Phyllanthus species, and curcumin and vithanilides are major compounds are steroidal lactones and glycosides, very well known as immunomodulators. These are all economically affordable and comparatively low toxic and very effective in chronic diseases. So here I have quoted some Nobel Prize winning concept in 2016 and 2018, where these concepts also directly, indirectly related to our traditional knowledge. So in 2018, Nobel Prize for the discovery for cancer therapy by inhibition of negative immune regulation, which is almost directly, indirectly related to our Ayurvedic concept Vata, Pitta, Kapha. Similarly, in 2016, for the discovery of mechanism of autophagy. Autophagy is nothing but fasting. So this is in Ayurveda, it is called Lankanalu. So that shows the importance of our traditional knowledge and traditional system of medicine. If you see different application, natural products are serving has its own applications in every field, starting from food, medicines, pesticides, cosmetics, and drugs. Here I have quoted very few which are very well known in the industries. First example, Artemisinin, we know pretty well. The discovery of Artemisinin rewarded as a Nobel Prize for Chinese-based professor Yeo Tutu, and which is very well known for the anti-malarial drug in the present market. And also Ivermectin B, very well known as a crop protectant, and rosmarinic acid, serving a lot in the as a food preservative in the present market, and Trapaxin B, very well known as a biological probe, and Epigenin is a very well known and hugely used as a artificial colorant, and Linolol is extensively used in the perfume industry, and stevia side is very well known as a artificial sweetener derived from Stevia rodendiana, and linoleic acid, which is a very well utilized in the perfumer industry in the majority of the lotions. And the recent use, this is a saponin called QS21, isolated from Colegia saponaria, and this is used as a very well-known adjuvant in different vaccines. Particularly, this every Covishield vaccine contains this adjuvant, which showed very high immune response. So that's what this has been, QS21 has been utilized as an adjuvant in different vaccines, particularly Covishield. So when you see the drug discovery aspect from the natural product, there is a different steps are involved, starting from bio raw material to drug candidate, where bio raw material can be different type of plant, fungi, insect, marine, and microbes, whatever it may be. So that has to be processed to get the extraction, and of course different extraction techniques are available, starting from Soxhlate, classical Soxhlate extraction to supercritical fluid extraction. So based on the secondary metabolites or compounds present in the plant, the all extraction techniques will be adopted to get extracted of all those compounds, and after getting extraction, to screen its the extract bioassociated fractionation is available. To get the pure compound, of course, chromatography techniques also, plenty of techniques are available based on the compound nature and compound polarity. All the chromatography techniques will be adapted, starting from the gradient chromatography, preparative chromatography, UPLC, HPLC, countercurrent chromatography, supercritical fluid carbon dioxide chromatography, membrane filtration. There are plenty based on the compound nature. All these techniques will be adopted. And after getting the pure compound for structural illustration, there are plenty, many spectroscopic techniques are available, and every spectroscopic technique has its own advantage to in the characterization of the compound, starting from UV visible, FTIR, NMR, mass, and finally X-ray. So every technique has its own significance in the confirming the structure, and the UV visible spectroscopy useful to identify the chromophores or double bond in the compound, and the FTIR spectroscopy useful for the identification of the functional groups present in the compound, and NMR is of course, NMR, proton NMR give the number of protons, and carbon 13 give number of the carbons present in your compound. And if you go depth in the 2D NMR techniques will give impression that the number of carbon-carbon attachment and carbon-hydrogen attachment, and mass molecular weight will be we will get from the mass spectrometry based on the different ionization techniques. That is also based on the compounds, the mass ionization techniques will be adopted. Finally, X-ray will give the complete structure of the compound. So once you get the pure compound, sometimes majority of the times these active compounds are scarcely available in the plants. So that will be developed a scale up and diversity and synthesis. So after getting the synthesis and diversification libraries, all these libraries will undergo for different clinical studies, phase studies, to get it as a drug candidate. So starting from first step to last step, it will take almost 10 to 15 years span if it is everything goes on well, and also very costly affair in the drug discovery project drug process from the natural sources. So when you see from the last 4 decades, the number of the natural product drug candidates evolved from nature to the drug. You can see from here I have quoted 1981 to 2019, almost 1881 drugs were evolved from drug candidates were evolved from nature, either a pure natural products or its derivative. Majority of the contribution goes to the anti-cancer drugs, where you can quote Taxol, Camptothecin, Phodophyllotoxin and its derivatives. And similarly, antibacterial drugs also evaluated the next percentage, and also several antibacterial drugs and other drugs. Even till today, the amphotericin B, which is called as a gold standard in antifungal therapy, is only origin is the natural source, that is isolated from soil bacteria or somewhat. And similarly, the lipopeptide, caspofungin, is also one of the important drug in the antifungal therapy, is also evolved from the natural sources. Even for dengue fever, even till today, Siddha medicine, papaya leaves are the best remedy to aggregate the platelets in the patient. That is a only source is papaya leaves. Similarly, if you go on speaking, and particularly in the pandemic period, pandemic also gave boosting to the plant-based medicines or immunity boosters. So like a C vitamin or immunity, ginger and all those things. So it has been highlighted the utilization of our traditional spices and traditional medicines to. So thanks to COVID for one side, not the other side. Similarly, the right side picture shows only 10% of flora has been utilized so far, and the remaining 90% the gold mine is as it is. So this is a good opportunity for the younger generations to take up the research particularly in the natural sources, natural product side. And when coming to fungi, 1 lakh to 3 lakh species are available. Only less than 1% has been studied till date. So if you see the laboratory approaches in the natural product research, there are different approaches, drug discovery approaches in the natural products area, particularly bioassay guided isolation procedure, bioassay guided approach. This is one of the classical procedure, and majority of many of the drugs has been discovered by adopting this procedure. Best example in 18th century, almost early 18th century, the morphine was discovered from Papaver somniferum, where that is the first drug which has been isolated from the bioassay guided procedure, and also that has also been evidenced, the starting from folklore knowledge or traditional knowledge to become a drug. So starting from that onwards, majority of the drugs has been discovered by using the bioassay guided isolation procedure. And the second one is the random approach, where you no need to follow the every step guided by the biology activity, and you isolate the as many number of compounds, and finally you submit screen for the activity to identify the active compound. And the NMR based approach, where this is also one of the recent approach, where you can also utilize NMR to isolate the compounds or to identify the compounds based on the NMR signals. And mass based approach, LCMS, UP-LCMS, MS, and hyphenated techniques will come in this approach, where to identify the molecular weight based separation and to fragmentation based separation to characterize the bioactive compounds. And metabolomics based approach also almost similar to the LCMS MS based approach. And molecular networking based approach, this is almost related to the artificial intelligence, where there will be some global server of natural products, and your system MS system will be coupled to connected to the global server. And when you develop the method, analytical method or fragmentation and molecular weights, that will be connected, since it is converted to global server. If it is a known compound, immediately you will get the information about those compound. If it is an unknown compound, then you have to work out for the purification, all those things. So we are all adopting all these methods for extensively for the isolation of the compounds from the medicinal plants, where utilized by the traditional people or folklore people. So when coming to our own research, we are working on in the area of basic and applied research, where in the basic research we regularly visit the interior forest and interact with the tribal people who has been utilizing the folklore medicines, and to get the knowledge or to get awareness about those plants, where they have been utilizing those plants from the several years and got the knowledge from their different generations. So when coming to the lab, we work on those medicinal plants, listed medicinal plant, to do whether it is already scientific validation has been done or not. So based on the literature, we will take up those plants to identify and isolate and identification of those active compounds. And further, we'll also do chemical modification or chemical simplification of that molecule to enhance and the activity and to reduce the toxicity, as majority of the pure natural products are somewhat not potent, and because of this, majority of the natural product based compounds or drugs in the present market are modified original natural products. So and further, after screening the activity, we will also do lead optimization to identify the active pharmacophore present in the molecule. So based on the active pharmacophore, we will go for further studies and activities. So these are the targets we are working: anti-cancer, anti-fungal, anti-diabetic, and free radical scavenging activity. In the applied side, we are actively involved in the standardization of the herbal drugs. Majority of the times we undertake these projects from the different herbal industries like Dabur, Himalaya, and other companies for the standardization purposes, and the process development of active compounds, bioactive compounds from the crude extracts. So this is an interesting and recent field where we do develop the process from the crude extracts without doing the classical column chromatography, and working in the liquid partition, precipitation, and different methods based on the compound nature and everything. So several other projects with collaboration with the industries and other government sponsored projects. So what are the challenges in the natural products research? Because natural products always contains a simple molecules to complex molecules, and these complexities of interpreting observed activities for crude extracts due to low concentration of the active compounds and potential antagonism synergism of multiple compounds. See, for example, in sometimes, if the active compound is a very minor quantity or picogram level, sometimes the activity of the active compound may be masked by the other compounds. So in that case, we will mislead. So that is also one of the biggest challenge, and interference of the some nuisance compound, because some natural product plant contains simple sugars and amino acids also. Sometimes this will mislead you to identifying the active compounds, so making it difficult to prioritize the leads for biosecure isolation. In increased cost for time consuming isolation, as you know, isolation natural products is a cumbersome process and involved tedious process for the isolation, and you need to purify the compounds fractions until you get the pure compounds. And structuralization is also process is also one of the biggest challenge and tough process. And you know, some of the medicines for camptothecin and podophyllitoxin in the olden days, in the I think in the 1800th century and 1700th century, for the confirmation of the structure, almost it took 15 to 20 years also for some of the complex structures. So that is a complex way. And reisolation of already known active compounds, which hinders the discovery effect, because the de-replication technique is also one of the important process for an important process to save the time, as it is the cumbersome and also time taking process. So in this we can utilize the analytical tools for the dereplication techniques, because new techniques like UPLCMS coupled to ESIMS have emerged recently for the identification and elucidation of the metabolites, secondary metabolites in the complex. What it will give you, just to prepare the extract and analyze, develop a method by using UPLCMS hyphenated techniques, and you can get all the molecular weights and molecular fragmentation pattern also you will get. So based on the fragmentation and molecular weight, you can analyze those compounds or simple compounds and the more complex compounds. Based on the fragmentation also, you can couple all the fragments to get the main compound. And even preparative also you can do by focusing some of the unknown compound. So I will share some of the recent results by adopting this UPLC hyphenated techniques for the isolation as well as the characterization by using the mass fragmentation of the mass fragmentation pathway, and by coupling the different fragments to get the structure. So we know pretty well this Qt of MSMS. This has the analyte spray and lock mass spray, isolation valve, and it's containing different dual stage reflection, ion detection system, high field pusher, transfer lens, and T wave collision cell, and quadrupole, and a lens and everything. So this is our recent work where we have isolated, we have identified different limonides from different two species called Cypadessa basifera and Xylocarpus granatum. These two plants belongs to Malaceae family. By we have identified different limonides based on the mass fragmentation using ESI Qt of MSMS, and also on their structural fragmentation study also been done. This is published recently in 2018 in JPBA. So before going to I would like to give emphasis on the limonides. The biological effect of the limonides include insect antifeedant and growth regulating antimicrobial. Limonides are nothing but our neem family compounds, and structurally limonides are formed by the loss of 4 terminal carbons on the side chain of apothiocholine and skeleton. That's what these are also called apothiocholine triterpenoids. And the limonides occur mainly Malaceae and Rutaceae families with 15 genera and more than 1400 species. Best example of Malaceae family is our neem tree, which possesses a diverse class of limonides, including azadirachtin, nimboloid, nimbin, jodinin, whatnot, everything, all famous limonides. Azadirachtin, we know, it's a very good insecticidal agent. So when you see this work, we have collected the plant and prepared the ethyl acetate extract and passed through the solid phase extraction cartridge, and then inject into the Qt of mass spectrometry to get mass spectrometer to get all molecular weights of the total ion chromatogram of the particular extract. And based on the fragmentations, we have identified these compounds, and I will give a different, and these are the fragments where we coupled to get this compound, identification of this compound based on the mass spectrometry principles, Mecklaffer rearrangement, retro-Diels reaction. and ester bond cleavage minus water, and these are all principles are well defined in the mass spectrometry. So based on this analysis, we have identified almost several limonides of different limonide class of compounds. So coming to sample preparation, we have collected the plant and extracted with hexane to remove all fatty material, and so that the material will be the free of fats, and then extracted with ethyl acetate. And since limonides contains are nothing but polar compounds, so that ethyl extract fraction will be the enriched fraction of limonides, and dried with centrifuge and evaporated at 45 degrees centigrade to ethyl acetate, and the dried extracts were solubilized in the acetonitrile and with the known concentration, and then passed through Phenomex the column, and I'm sample injected into the ESI Qt of MS to get the total ion chromatography. These are the mass spectral conditions we have optimized. Mode top ESI positive mode, mass range 50 to 1200 MZ, and capillary voltage for fragmentation 3 kilowatts, and source temperature 120 degrees centigrade, dissolution gas temperature 350, and all these validated and optimized conditions were set up, and after that injected to get the total ion chromatograph of this extract, Xylocarpus granatum. And then here we gave some compound names also. These are the structures of the compounds, and we have identified based on the molecular we have identified based on the molecular fragments. And the next when next, similarly, these are the proposed fragmentation pathway for proposed in the limonides. So these are the, so here you can see we can see from here the ester bond cleavage. This sodium adduct has been formed. This molecular weight is 525. We can observe, and through McLaughlin rearrangement, this fragment has been formed. Its molecular weight 287. Here you can see all the fragments. Similarly, from this also minus H2O and minus acetic acid, and all these fragments here you can see. Based on this fragmentation pattern, we have confirmed all the structures. And similarly, this the other type of limonides also observed. Here we can see the major molecule 505, and the other fragments also you can observe here. So based on the analysis and the of these fragments, the structure has been confirmed. Similarly, the other from other plants, Cypadessa bassifera, which is also belongs to Malaceae family. The total ion chromatogram, from the total ion chromatogram, we got all the molecular formulas of the compounds, constituents, and based on the fragmentation pattern, we have identified the structures of all these compounds from the fragmentations. Here we have proposed one or two compounds of the fragments, like which has been formed retro-ene reaction, 1,3-H rearrangement, McLaughlin rearrangement. These are all very well defined in the mass spectrometry. Here also I proposed some Mexicanoid limonide type compounds, where we have identified the compounds based on the double bond shifting followed by the retrodyl salt reaction, minus H2O, minus CO2, and all those things. So conclusion, this is a fast and simple method was developed for the accelerating dereplication studies on limonides. In the analysis of these two plants, Cypadessa bassifera and Xylocarpus, we are able to identify the 25 limonides using the MSMS fragmentation pattern. So similarly, this is another plant where Trichilea corneroides. This also belongs to Malaceae family, and this plant has been chosen based on the interaction with the tribal people, particularly in the Rampachodavaram region, where we came to know this plant has been utilized by the tribal people as a folklore medicine for different applications, healthcare benefits, as well as this plant is also using as a agriculture purposes. So we have been chosen this plant and prepared the extract. and applied the LCMS guided strategy for the isolation of the compounds. What we did initially, we prepared the extract, and then the extract has been injected to Q of MS to generate the total ionic chromatography and generate the molecular formula and molecular weight. And based on the molecular formula, molecular weight, we searched in the dictionary of natural products as well as reactions to identify those known compounds and unknown compounds. When unknown molecular weight has been observed, that has been pinpointed to go for a preparative HPLC and fractionation and preparative HPLC to for the purification. Finally, by using NMR, all the structures has been confirmed. These are the compounds, 6 compounds which has been isolated and purified by using the LCMS guided isolation. And for a glimpse here, I gave the proton and C13 NMR for the compound 1. So these are all new compounds and reported in published in Journal of Natural Products recently. So another work, the lichens, where we have taken the 5 species of different lichens and the abundant lichen, we have isolated the compounds by using the chromatography. By using those pure compounds as a standards, we have mapped these compounds, presence of these compounds in the different lichen species by using UPLCMS technique, and also we have quantified by using this technique, MRM multi-reaction monitoring method. So this is a workflow where we have collected the species and MSPD extraction, comparative analysis, and confirmed the lichen substances. So before going to the work, just I would like to give the importance of the lichens. Lichens are nothing but the symbiotic organism of fungi and cyanobacteria. And from this we have collected this five lichens particularly in the Arsley Hills and Rayalaseema region, Permotrema, and here I have identified properly by using spot test and the chemical methods and taxonomy and other methods. And here the Permotrema tinctorum is the abundant lichen. From that lichen we have isolated these 5 compounds, and these 5 compounds were utilized then as a standards to map these compounds, presence of these compounds in the other lichens, because the lichens are also known as a pollution indicators, where this, when the lichens are grown in that particular area, that particular area is a non-polluted area, then only lichens will grow. So when you collect these lichens, minor lichens are non-abundant lichens, so it will take another 5 to 6 years to grow. So to avoid the disturbances in this chemical ecology, we have developed the analytical method for the mapping and quantification of these compounds in the other lichens. So initially we developed the TLC pattern of these lichens and with the standard compounds also. So for that, initially we prepared, we have developed the method by choosing different extraction methods, starting from cold extraction, ultrasonication, MSPD, non-polar to polar solvent washing with different solvents, and on. Finally, the MSPD extraction is giving good yield. So we have optimized the solvent also, acetonitrile. So with this optimization condition, then we went off, we moved to UPLC Q-TOF conditions. Here we have optimized chromatographic conditions as well as mass spectrometry conditions. With these optimized conditions, we have developed the total ion chromatogram of all the 5 species of Permotrema species, and identified the molecular weights and molecular formulas by doing the MS fragmentation, and identified almost 47 compounds from by using the fragmentation of pathway. Here I have right side, I propose the fragmentation pathway of this class of compounds, and similarly here also I have proposed the structures of the compound, and same explain the fragmentation pathway of these compounds, how they have been formed, all the fragments, how we have been coupled based on the mass spectrometry principles. So similarly, the monophenolic compounds also identified. This is a fragmentation pathway we proposed based on the fragmentation peaks observed in the mass spectra, and paraconic acids also observed in the plants, in the lichens, and fragmentation pathway also proposed here. So finally, we concluded by using the Venn diagram the distribution of the compounds in the 5 species. So in the almost a few compounds are distributed in all the species, and some of the compounds are restricted to only few species. So the conclusion of this this work, optimization of the MSPD extraction lichen for the first time, and the UPLC MSMS method, developed a UPLC MSMS method, helped us to identify 47 compounds, including 9 new compounds, and also comparative analysis of Phermotroma species by using this species distribution also been done by using this UPLC MSMS. And the another lichen work where we have used this red color lichen, top lichen, and extracted and then injected in the UPLC MSMS to get the molecular formula and molecular weight of this each and every compound. And based on this, we have identified the purification and then identified the new compounds here. I have given here, and all these new compounds were also tested for different activity where they have been displayed moderate activity against the tested cell lines. So this is another work where we have quantified and standardized and also verified the different presence of this boswellic acid in different Boswellia serrata extract as well as different products in the market by using UPLC Qt of MSMS. Boswellia serrata is a very well-known Indian frankincense, and it has been used for in the Asian and folklore medicine for the treatment of arthritis. And this is called in the traditional medicine as a shallaki. This resin is being used widely in the herbal formulation for different Ayurvedic formula in India. These are the 6 markers available in the Boswellia serrata. So if you are using Boswellia serrata as a formulation, definitely the 6 markers should be present. So what we have done, we have collected the material, and we have collected the material and quantified the 6 boswellic markers in the Boswellia serrata, and then by developing our own method, which is almost on par with the European pharmacopoeia and US pharmacopoeia, and then moving ahead, we have procured several of the formulations like powder formulation, cream and tablet, and multi-herbal formulations where the which contains Boswellia serrata gum. So, and analyzed for the presence of the 6 markers, and by these are the chromatographic and mass spectroscopic conditions, optimized conditions, and developed the total ion chromatogram of different Boswellia extract samples, marketed formulations, powder, cream, liquid, multi-herbal formulations, and quantified all these boswellic acid 6 markers in every sample. And however, majority of the samples are not containing boswellic acid boswellic acid markers, and we are very surprised after seeing these results. Very few formulations contains these markers. So this is also one of the standardization aspect of the Boswellia formulations. So further moving ahead, we also analyzed the minor compound present in the boswellic acid extract and identified through the fragmentation pathways, and we have identified 2 or 3 new compounds also. So this is the quantification of different formulations like a capsule, cream, powder, and tea fusion and gel, everything. Here I have just mentioned the values. And tentatively characterized compounds from Boswellia serrata based on the fragmentation pathway, we have identified these compounds in the extract. So finally, this is a simple and UPLC PDA method developed for the quantification of 6 markers in the Boswellia serrata gum extract, as well as in various commercial formulations. And this technique was applied for the characterization of the metabolites, including boswellic acids and thirucalic acid in the Boswellia serrata extract. MSMS fragmentation of boswellic acids and thirucalic acids would be a valuable tool for the quality control assessment of commercially available formulations containing Boswellia serrata. So when coming to quality control aspects of herbal drug, we know the therapeutic effect of any herbal drug or any medicinal plants are due to the presence of the biological active compound, which are mostly secondary metabolites like alkaloids, flavonoids, terpenoids, coumarins, terpenes, etc. So for the herbal formulations, majority 75 to 80% of the medicinal plants required, the traditional medicine is collected from the wild source. So sustained supply of quality raw material is therefore difficult. So in that case, the adulteration is happening. So similarly, the medicinal plants used in the traditional medicines are cross-pollinated species, and these are all sometimes the plants also the variety of the factors, genetic factors, and macro environment, season, and stage, growth, development also influencing the plant. So if you collect the raw material in one season, and if you collect in the summer season also, that affects the existence of the compounds in the plant. So height and environment, as well as the season also influence the compound. For example, if you prepare the chawanprash, or if you prepare the any extract from one plant in the Bhimavaram region, is different from the Nepal region as well as the Himalayan region. As in the Himalayan region, because if it's cold environment, the plant the constants may be different from the Bhimavaram region, because oxygen supply is low in Himalayan region when compared to the Bhimavaram region. So these are all different factors influence the existence of the compound that has to be standardized before using into the formulation. So lack of consistency in quality in batch to batch production of the products is also one of the aspect for the standardization. Lack of a well-defined and well-orchestrated standard operating procedure is all. So, in standardization of the herbal medicines, the parameters, quantity, quality, adulteration, and efficacy. These are the different parameters has to be checked in the standardization of the herbal drugs: physical, chemical, biological, and botanical. In the physical parameter, moisture value and ash value, extractive value has to be checked out. And chemical, the raw material should be free of pesticidal residue and free of heavy metal residue. And in biological, the raw material should be free of microbial contamination, and its pharmacological value and toxicology evaluation should be done. In the botanical, macroscopic and microscopic observation should be, or evaluation should be done, because in some of the herbal companies are, when the original plant is scarcely available, they are using the synonymous plant of the original plant. So in that case, the microscopic and macroscopic examination should be thoroughly done to identify the synonymous plants. So these are the analytical techniques well approved by US FDA and other agencies: HPLC, HPTLC, UPLC, LCMS, and GCMS. Every technique has its own significance for the characterization and identification of the compounds. These are well approved techniques in the US FDA for the standardization of the herbal medicines. So just here, this is one of the example where we have done the standardization aspect of plant is Asurya, which is a very well-known plant used in extensively used in the Ayurvedic formulations. This plant exists in the northern part of our country. So these are the dihydrocortisolactone, cortisolactone are the two marker compounds. So what we have done, we have procured the plant material and quantified these two markers in the raw material by developing a HPLC method. And further, we purchased some of the commercial formulations of different companies, different vendors, and we have by using our method, we just cross-checked whether these formulations are containing these marker compounds or not. So fortunately, all these formulations are containing these two marker compounds in the detection limits. Further, we also studied the stability studies of these marker compounds, because if the marker compounds are not stable, because as our intestine contains acidic environment, if it is not stable in stable acidic environment, it may cause the some adverse effects. So we also studied the stability studies of these two compounds. Fortunately, these two compounds are stable enough in the stressed environment. So this is another hyphenated technique, HPTLC. We have coupled with the QTOF MSMS technique for the rapid confirmation of and fingerprinting analysis of lichen compounds. So here we have coupled the TLC MS interface to the QTOF MSMS and analyzed the 3 lichen species to identify the compounds. We have isolated and we have identified and did the preparative HPTLC MS for the identification of those compounds. This is a mobile phase we have used for get the better separation. the TLC pattern of these three species. And these are the compounds we have identified by using the hyphenated HPTLC MS and QTAF MS technique. And this is the method is very simple and time consuming, little less time consuming. More number of samples can be analyzed on a single HPTLC plate with better efficacy and accuracy, and efficient tool for fingerprinting the plant extract as well as the lichen extract samples in comparison with the standard based on their RF values. The hyphenation of the mass spectrometry provides the simultaneous separation and identification of the major analytes present in the samples. In this method, the hyphenation with the QT of MS provide the exact mass of analytes with which elemental composition can be generated. So overall, the role of, if you see the role of natural products in the present in the today, nature still holds many secrets which can learn from the chemical structure, biosynthesis, and the role and relationship of the plants to the other organs. The study of the natural products merges with the science, with culture. For countries harbor rich biodiversity, the study of natural products is an important way of developing the natural resources. Of course, natural products should use the new developments like a hyphenated techniques and dereplication techniques, molecular biology, computational science to speed for the speedy isolation and identification, and to get the more new bioactives from the extracts. So these are the different various Indian scientists who spent their entire life in the natural product research, starting from J.L. Simonson, Ramnath Chopra, Shilamuddin Siddiqui, T.R. Sheshadri, K. Venkatraman, T.R. Govindachari, Azima Chatterjee, Sushanka Chandra Bhattacharya, and Sukhdev. He is called as a living legend of the natural products, and he is the father of called steroids, and almost T.R. Govindachari is known for neem scientist because he majority of his entire life, most of his entire life dedicated to isolate and identification of the compounds from the neem Azadirachta indica. And Asima Chatterjee is also very famous for her contributions in the isolation identification of the various alkaloids in different medicinal plants. So finally, I would like to thank our the former director, Dr. S. Chandrasekhar, and Dr. J.M. Rao, who is my supervisor. Because of him only, I was in this unique field of unique area of natural products. And of course, my students, because of their hard work only, I was able to show all these structures and all this work. And not least the funding agencies, because of this their financial support only, I have carried out all these research. And thank you once again for your patience, and also thanks for giving the opportunity to showcase our recent work for utilizing the hyphenated techniques in the isolation of the bioactive compounds. Thank you one and all.
Sir, thank you, sir. Sir, please open your video, sir.
Yeah.
Thank you very much, sir. Thank you for sharing your research to the participants. So then the topic is open for discussion. Any participants likely to ask a few questions to the today's speaker, sir? Please unmute and ask the questions. Participants, please unmute and ask the questions. Participants, please unmute and ask the questions. Okay, sir. Sir, I have one question, sir. Meanwhile, the participants will ready to ask the few questions.
Please.
Sir, you showed in the laboratory approach, several approaches you showed, sir, in which NMR based approach, maybe it is difficult task, sir.
It is a recently evolved technique, sir. By using NMR also, we can identify the mixture of compounds in the extract.
Okay, sir. And because of the even the proton NMR, C13 NMR.
Now, based on the proton NMR only, in that case, we only focus on the characteristic peaks of the compounds.
Yes, sir. Generally, when you recorded the NMR spectra, Suresh Babu. Sir, are you audible, sir?
Yes.
Generally, when we are recording the NMR spectra, that is somewhat complex compounds are difficulty to characterize.
Yes.
If more than one compound is there in the bio-guided fraction, spectra will be very lengthy, sir.
Very lengthy, and we can identify number of compounds also.
Okay, that is by experience and skill only you can able to.
Yes.
Thank you, sir. So, any guiding principle we have any literature or any book using the MS and the NMR to identify the guided active bio-guided fractions from the natural
I
products?
think
Sir?
books are available, but these are all whatever I have shown our own research results.
Okay, this is your own research.
Own research results. You will never find, of course, majority of them are published. You can find the articles, but these are all not part of any textbook and any computer like other things.
Okay, sir. Sir, one of the participant is likely to ask some question.
Yes.
Sir. Hello, Suresh Bobosa garu. Participant Suresh Bobosa garu. Please unmute and ask the question. Hello, Suresh Bobosa garu. Please ask the question.
Sir, I'm Divya sir from Sri Vishnu College of Pharmacy.
Video. Please ask the question.
Sir,
Please ask the question. Sir is waiting.
sir, what are the main advantages of hyphenated techniques in analyzing secondary metabolites, sir?
Yeah, that's what I have shown. You can use the hyphenated techniques to reduce the time, and as well as a dereplication technique also you can use by LC-MS-MS and UP-LC-MS-MS. Analytical techniques as playing a major role, particularly in the herbal drugs standardization and identify the active compounds and analysis of the secondary metabolites.
Anybody like to ask the question?
Good afternoon, sir.
Please tell me your name and college name.
Good afternoon, sir. Myself, Kavya. I'm Sri Vishnu College of Pharmacy.
Yeah, good afternoon.
Can we use electroanalytical methods for analysis, sir?
Sorry?
Kavya,
Can we use
please ask
electroanalytical methods, sir?
remove the mask first.
Can we use electroanalytical methods for the analysis, sir?
Yeah, you can use.
Okay, sir.
Good afternoon, sir.
Yeah.
Myself, Lakshmi Vilka Bhavani. I'm working as assistant professor here, sir. So
Okay.
I'm having small doubts, sir. Which type of difficulties we are facing during analyzing of natural products by using hyphenated techniques, sir? Means if there is any other troubleshoots or any difficulties, means while by using in case of interference we are using sir. At that time we are facing any difficulties.
Yeah, it's all depend upon your skill for the isolation, for the separation of those compounds. If you can develop a method in such a way that nice separation should be done, should come, then only you can analyze each and every peak by application of the hyphenated techniques.
Okay, sir, another small doubt, sir. In some research point of view articles, I listened that
Nowadays, they are using medium pressure liquid chromatography for isolation of natural products. Means either it is advantageable or
Yeah, that's
is it-
what for any chromatographic technique will be adapted based on the compound nature.
yes, sir.
Yeah, for example, if it is alkalides, then you should use the neutral alumina because
Yes.
alkalides are base in nature and silica gel is acidic. So if you use normal silica gel, the compounds will be binded to the silica gel. You will not that those are not eluted. So based on the compound nature, all the techniques will be adopted. That is also one of the important factor in the natural product research for the isolation.
So that's why we can select the medium pressure liquid chromatography, sir, based on
Yes,
the nature.
yes.
Okay, sir. Thank you, sir. Thank you for giving a good lecture.
Thank you.
Audience, please, any questions from the audience? We will like to conclude the session. Please ask one or two more questions to the today's speaker, sir. Participants, please unmute and ask the question.
Good afternoon, sir. I'm Rani from Sri Vishnu College of Pharmacy. My
Yeah.
question is, how often the metabolic studies are used?
Metabolic studies.
Metabolic studies.
Yeah, metabolomics is a very hot area in present scenario. That will help you particularly in the to assess the quality control aspects of herbal drugs.
Thank you.
Okay, thank you, sir.
Thank you.
I would like it to conclude for the today's session by brief closing note. First of all, I would like to say thanks to Dr. K Suresh Bobosa garu, Principal Scientist at the well-famous CSIR IICT Laboratories, Hyderabad. Thank you very much, sir, for spending the more time in our busy schedule and sharing your knowledge, especially in our research area, and explaining your total research experience, recent research experience from our laboratory. Sir also explained the importance of traditional medicine in India and other countries. And along with that one, the sources of traditional medicine also explained by the Suresh Babu sir, and various applications of natural products also discussed to the participants, and also explained how to convert bio-raw material into the drug candidate. That is the one of the most important laborious work in the natural product chemistry. Sir showed very simple way how to get the raw material into the drug candidate. Sir also explained about on the isolation of the various varieties of the limonites, lichens, boswellic acid and their derivatives from his laboratory discovery. Thank you very much, sir, for sharing your views to the participants, and thanks for all the participants for grand success of the second day of the 3 days national webinar conducted by the Sri Vishnu College of Pharmacy. Thank you very much, sir. Thank you, sir.
Thank you, sir. Thank you very much.
Thank you, sir. Nice meeting you, sir.
Yes,
Bye.
sir. Thank you.